Every day, we encounter millions of infectious agents through the air, our food, and the surfaces we touch. Yet we rarely fall ill. This happens because the host body possesses an innate ability to fight off most disease-causing organisms, an overall defense mechanism we call immunity. To see how this system works, let's track a specific pathogen trying to invade the body, Salmonella typhi, the bacterium responsible for typhoid fever. As Salmonella attempts to enter and multiply, it faces a two-tiered defense system. We divide immunity into two main categories: innate immunity, which is present from the time of birth, and acquired immunity, which develops over a person's lifetime. This division shows how the body manages threats. It relies on an immediate general shield to block initial entry, followed by a highly specific counterattack designed to neutralize whatever slips through. This diagram outlines innate immunity. It is a non-specific defense mechanism that uses 4 distinct barriers to stop pathogens like Salmonella typhi before they can establish an infection. The first obstacle is the physical barrier. Skin is the primary wall preventing microbial entry, while the mucus-coated epithelium lining our respiratory and gastrointestinal tracts acts like flypaper, literally trapping microbes before they can go further. If Salmonella makes it past the physical walls, say through contaminated food or water, it hits the physiological barriers. Acid in the stomach, along with lysozyme enzymes in our saliva and tears, creates an inhospitable environment that actively destroys microbial growth. Should the bacteria reach the tissues, cellular barriers step in. White blood cells like macrophages and PMNLs engulf and destroy the invaders. Meanwhile, if a virus is the attacker, infected cells deploy cytokine barriers, secreting proteins called interferons to protect non-infected neighboring cells. Let's test your understanding. Based on these 4 categories, where does the stomach acid that neutralizes contaminated food belong? It is a physiological barrier. By constantly deploying these 4 roadblocks, the innate system limits the spread of infection, buying the body the time it needs to activate its pathogen-specific defenses. This chart shows what happens when Salmonella survives those initial roadblocks. The body's second line of defense takes over, acquired immunity. The first encounter with a pathogen generates a primary response. It is slow and of low intensity, but it allows the body to build biological memory of the attacker. Because of that stored memory, any subsequent encounter with the exact same pathogen triggers a secondary or anamnestic response. The body remembers the invader and mounts a rapid, highly intensified counterattack. This response relies on specialized white blood cells. B lymphocytes act as factories. When they detect a threat, they produce an army of specific proteins called antibodies, releasing them directly into the bloodstream to fight the pathogen. Because this happens in the blood, we call it the humoral immune response. T lymphocytes operate differently. They do not secrete antibodies themselves. Instead, they act as a surveillance team that scans for cells that shouldn't be there, including human cells hijacked by viruses or foreign cells from another individual entirely. By identifying these foreign signatures, the cell-mediated response allows the body to differentiate its own tissue from non-self intruders, which is why it will identify and reject a transplanted organ as a foreign object. By dividing the labor, using B cells to manufacture weapons for the blood, and T cells to manage infected or foreign cells directly, the acquired immune system can neutralize complex, highly evolved threats. To understand how these weapons work so precisely, we have to look at the physical structure of the antibodies produced by the B cells, represented biologically as H2L2. Every antibody molecule is a Y-shaped structure built from 4 peptide chains. It consists of 2 longer inner segments called heavy chains, the H2, and 2 shorter outer segments called light chains, the L2. The extreme tips of this Y shape are the antigen binding sites. At a molecular level, the protein chains are folded into a unique geometric signature. This physical shape matches a corresponding receptor on the surface of the Salmonella bacterium, allowing the antibody to dock with and neutralize that specific invader while ignoring everything else. Because the body can generate specific H2L2 structures for different invaders, it can capture and neutralize exact threats based on its biological memory. We can leverage this system in 2 ways. Active immunity occurs when the host is exposed to antigens, either through natural infection or deliberately via vaccination. The body slowly produces its own antibodies and builds a lasting supply of memory cells. Passive immunity provides immediate protection. Instead of making them, ready-made antibodies are introduced directly into the body. For example, a mother passes IgA antibodies to her infant through colostrum. This provides instant defense, but the infant's body does not produce memory cells from this process. Let's map exactly how these systems work together. We have innate immunity, our immediate nonspecific shield built on physical, physiological, cellular, and cytokine barriers. And we have acquired immunity, the specific memory-driven response powered by B cells in the blood and T cells in the tissues. Mapping these pathways allows modern medicine to step in. By using vaccines to train our B and T cells safely, we can replicate and boost our biological defenses, equipping the body to win the invisible war against disease.